Hi guys, this is just going to be a pretty fast summary of Chapter 6. Just looking at big picture things and mostly understanding how light moves through the retina and through the brain. So light is a visual stimulus. It's a wave that varies in amplitude, length, and purity, and it's measured in nanometers as you can see by the image above. And light is processed through our eye, of course. Make sure you know the function and location of each part of the eye, like the cornea, iris, pupil, lens, and retina, but I'll mostly be focusing on the retina. And all of these parts of the eye are critical in the process of sensory transduction, which is the conversion of sensory stimulus to neural activity, and this occurs at the retina. So I'm going to start by talking about the photoreceptor layer of the retina. The two very important parts are rods and cones, and we have a lot more rods than we do cones, but they're distributed differently. So for example, there are a bunch of cones in the fovea, but no rods, and a lot of rods in the periphery, but not many cones. So it's also important to know that the cones are meant for bright light and color detection, which creates a pretty sharp, precise image. And then whereas the rods are meant for more dim lighting and create a duller image. The way I memorized this was thinking about the pointy edge of the cone as being sharp, like a sharp image, and then the flatter, more dull end of the rod being a dull image. That kind of just helped me a little bit. And then the next component is the lamellae, which are thin layers of membrane in the photoreceptors that contain photopigments. And those photopigments are consisting of opsin and retinal. Opsin for color sensitivity and retinal for light photons. Alright, so sensory transduction. This is just a quick overview of the process, but when light hits the retina, photopigments are activated. They then split into the opsin and retinal. And the receptor potential, which is similar to membrane potential, changes because of this splitting. So if you think back to what we've already learned about membrane potentials and how a change in the potential will cause change in the release of the neurotransmitters, it's the same concept. So in this case, the neurotransmitter is sent to a bipolar cell, which then changes the activity in this cell and results in a change in how much neurotransmitter the bipolar cell releases. Next, this bipolar cell is going to communicate with a ganglion cell, affecting the action potential that this postsynaptic ganglion cell sends, and then the axons of this ganglion cell are going to form the optic nerve. So the takeaway is understanding how light can influence the receptor potential and signaling of these cells, which leads to the transfer of information to the back of the brain and occipital lobe. An important thing to also understand is the backwards organization of this process, because of the epithelium. And the epithelium would be located on the right side of the image between the back of the eye and the photoreceptors. And the photoreceptors have to be next to the epithelium because it provides them with all of the nutrients that they need to function. And so the only problem is that the epithelium is opaque, meaning that light cannot pass through it. So in order for these photoreceptors to get light, the light needs to pass through the bipolar and ganglion cells, which are transparent first. That's why it's backwards. Alright, and then these next few slides I'm not really going to go in depth, but make sure you read the textbook and watch the lectures to know about midget bipolar cells and diffuse bipolar cells and how this creates a difference in imagery. Same with color perception. Just know all of this information. Talking about opponent process coding. Different ganglion cells are excited and inhibited by different colors in different areas. Same with receptive field. Make sure you know about on cells and off cells. Alright, so let's focus on the primary pathway that light travels to get to the back of the brain. This is the retinogeniculocortical pathway. This starts at the retina, goes to the lateral geniculate nucleus, and then to the striate cortex. So starting from the point where the axons are forming the optic nerve, from here the signal crosses over at the optic chiasm, and next the optic tract travels to the lateral geniculate nucleus, which is in the thalamus, and then to the primary visual cortex or the striate cortex or area V1 in the occipital lobe. And then the LGN has 6 layers. Each different layer has different size of cell bodies and different information is processed there. So there's the magnocellular, which are larger cell bodies in layers 1 and 2, and the parvocellular, which are smaller cell bodies in part 3 and 6, and then coniocellular layers are just the smallest. And then the next part of the process involves the striate cortex, which also has 6 layers, and this is involved in the process of pretty basic visual information such as line orientations, movement, spatial frequency, and color. Just a brief overview of the cells in the striate cortex. There are simple cells, complex cells, and hypercomplex cells, each meant to process different types of information, and these function through excitatory and inhibitory regions. So for example, in simple cells, a cell will become excited when a line falls directly in the excitatory region, as well as become inhibited when the line falls in the inhibitory region. And this would help us with information about things like edges. There are also different modules, which are independent, self-contained, and each module processes one area of the visual field. And each module has its own organization within itself. There are CO blobs, which detect color, and then everything outside of the blob detects everything but color, so like motion and orientation. So then from the striate cortex, or area V1, the information is integrated by the visual association cortex, also known as the extrastriate cortex, or area V2 and above. This is generally how the information is fed, but in some cases information can be fed backwards from areas V2 and above to area V1. And then the last important component to cover is the 2 different streams of analysis, the ventral and dorsal streams. Information goes from the striate cortex to the extrastriate cortex and then travels through either the dorsal stream towards the top of the head or the ventral stream towards the bottom. And the ventral stream is meant for information about form, color, and this comes from magno and parvo cells, otherwise known as what information, that functions to identify what objects are. And then there's the dorsal stream, which is for information like motion and location, mostly coming from magno cells. And this is why it's named the where or how stream, like where objects are or how to interact with the objects. So finally, I know I didn't really cover much about color, but I did find this study that I found pretty interesting that talked about color and the mood of college students. So I just added this snapshot of the aim of the study, and if you want to go read up on it, here is the reference down there.